Greetings. Today we're going to discuss separation of mixtures. Let's remember that mixtures are of two types, homogeneous and heterogeneous. Homogeneous mixtures are the same throughout. Heterogeneous mixtures are not. Now, there are many techniques by which we can separate a mixture. If you take a look at this slide and look at the yummy tasty M&Ms, we know that we can separate them easily by simply picking out all the green ones or all the yellow ones or all the red ones, and that way we will have separated effectively our mixture. But we have to think about other mixtures that are a little bit more complicated to separate and what these techniques are actually called. The key to separating mixtures is to be able to identify a difference and then exploit that particular difference. We can certainly realize the difference between this elephant and the little mouse because if they are both outside of a house and the mouse wants to get away from the elephant, the mouse can easily go into the house through any little crevice, but the elephant cannot. So you will have effectively separated them. Let's remember also, before we talk about separation, that separation does not chemically change substances. These techniques only work for mixtures, not for any other substance. All right, so let's take a look at this first technique, manual separation. Well, this is pretty easy. This is all you have to do is to separate each substance, just like the M&Ms. We can take the jelly beans, and if I only like green jelly beans, I would separate the green jelly beans by hand, pick them out. On the other side, we have somebody separating the little shafts and the little dirt and the little stones from rice. So that's easily done because you can actually see those particles. They're pretty easy to see and pick out. And if we couldn't do it by hand, we could do it with a little forceps or tweezers or something like that. The next separation is by the difference in magnetism. Well, if I have two substances that are mixed into a container, and I have one that is magnetic, the other one that is not, easy. I can just take a magnet, stick it in there, pull it out. All the magnetic stuff will come out with the magnet, and the rest of it will remain. The stuff that's not magnetic. Say you had a mixture of rice and little pieces of iron, okay, as in cereal, okay. Maybe we could separate the cereal from the iron from the cereal. So it's easily separated because these two have two different properties. In this picture, it shows sulfur and iron being able to be separated because one is magnetic, the other one's not. A separation you may be more familiar with if you make coffee or drink coffee, it's filtration. That's how we make coffee. We use a coffee filter. Well, in this case, we're not using a coffee filter, but in this case, we're using a funnel with a filter paper inside, which is folded in a special way, which I will show you in the lab. And you separate these substances by the difference in particle size. The particle sizes separate them because the larger particles are trapped in the funnel and the rest of it goes right through. And the stuff that goes through is called the filtrate. And again, another separation technique, filtration. Fairly simple. I've put two in the slide. It's evaporation and distillation because they both make reference or they act on the difference in boiling point. The one on the left, we don't care so much about one of the substances. We care about only keeping the second substance. So we evaporate it and As in the case of water and salt, the water would go into the air, the water would evaporate, and the salt would remain behind. On the other hand, on the right side, we have distillation technique. This is used to separate liquids that are mixed together, but that they have different boiling points. This is a technique that's used quite a bit in the petroleum industry to separate all of the um, various compounds that are part of the of petroleum, of crude oil, and eventually it's distilled, and uh, all the substances that are organic compounds that can eventually be used to separate. I'm sorry, to use in medicine and cosmetics and gasoline and jet fuel, and all these different substances can be separated this way. We have a distillation apparatus in the lab that I will show you. It's in the back in the stockroom. And what we do there is we actually distill the tap water. Tap water is not pure water. It contains a lot of chemicals, other chemicals in it, including magnesium and chlorine, which is added for cleaning it or for killing bacteria, and many other minerals. And it is separated by distillation, and it comes out as pure water. Another thing that can be used for distillation is to separate two liquids that are miscible in each other. That means they dissolve in each other, and they can be separated by the differences in boiling point. As the boiling points are reached, the first one that is distilled out is the one with the lower boiling point, and then the next boiling point is distilled next, and there's a plateau in temperature as the distillation points or the boiling points are reached. Another technique is precipitation, and this depends on differences in solubility and density. On the left-hand side, we have a centrifuge. What a centrifuge does is a machine that spins very fast. You place test tubes inside of it, and as it spins fast, it separates the materials into layers, usually 2 layers, and it's used to spin blood and to be able to separate the different components of blood. The denser components settle to the bottom, and the less dense components, usually the plasma, is at the top. And this can be used then to study the blood and to do various tests. The one on the right, on the top, is a precipitation that has taken place, but in this case, the density is such that it did not need to be spun. It is has separated on its own, and then you can decant. You can pour out the liquid, and the solid is left behind. The same principle is used for the bottom. If you see the bottom right, there's what we call a separation funnel or separatory funnel, and it has 2 layers, and you can open the little stopcock at the bottom, allow the fluid to the blue fluid, the lower one, to flow, to seep out, and then you have the other substance left behind. Sometimes the same decanting can be achieved simply by pouring it out of a beaker, and usually it flows down a stirring rod, and it works fairly well. Finally, it's chromatography, one that you have probably used before, possibly, if you've made a tie-dye shirt. It basically has, depends on separation by how substances, differences in the substances between a mobile and a stationary phase. The stationary phase in this case would be the ink that is separating, and the water is what the mobile phase, the one that moves up. Here we see separation of ink in soluble inks in water. So